Hello, I am Dane Parker, and I'll be describing what DNA is using this model. First, deoxyribose nucleic acid. That's basically what DNA is. It's another word for DNA, and what DNA is used for is the transportation and information of cell growth, and it's used to grow your body. It's how you grow and it tells you what you look like. I'm sure you already know that, though. A macromolecule is a large molecule split up into four groups. These groups are nucleic acid, which is used in the substance that is used in the formation of DNA. Proteins, lipids, and polysaccharides, however, have little relevance in DNA. Next, the monomer of DNA are called nucleotides. There are four different nucleotides: adenine, guanine, cytosine, and thymine. And basically, as you can see, those are these things: the GA, TA, and then there's C over there, as you can see. I'll explain those a little bit more later, but for now, the sugar polysaccharides are the backbone that forms the structural framework of nucleotides. So basically, do you see the popsicle sticks? That's kind of like, it holds it all together. Like, it's what groups the ATs and adenines, and thymines, and cytosines all together and keeps them in one place. Next, deoxyribose sugars are phosphate groups to form the backbone with DNA. So basically they work with phosphate, and they also form the backbone of DNA together with phosphate. And now I'm going to go back to these groups here, the ATs and the Gs and all the stuff you see down there. There are four nitrogenous bases, as I stated before, found in DNA. They are called guanine, adenine, thymine, and cytosine. They are basically told, I'm going to just keep calling them by their first letters from now on. So whenever I say A, it's adenine, T for thymine, and so on and so forth. The bases can be divided into two categories: pyrimidines and preamines. And in the pyrimidine group are thymines and cytosines, and in the preamines, sorry, I can't really say that too well, is adenine and guanine. These bases are responsible for DNA replication. When DNA splits into two bases, these bases will split apart and make new DNA. So let's say this A splits off. Then the T on the other side, over there, as you can see, will split as well. And so the DNA, how it knows what's on the other side is what, like, if it's A on just half of a DNA strand and it wants to make a new cell, it's got to know what's on the other side. It's like drawing half a picture. Since it has one half of the DNA, it knows what the other half is. Sorry for this part here kind of being in the way, but I did my best with this whole creation thing. Hope you can see the T under there pretty well. If you can't, there's one right behind it. It's basically the same thing. It basically goes all the way down to the bottom of DNA. Usually DNA would be much bigger than this, but as I said, not that good of a crafter. So yeah, these bases are also what allows you to copy DNA. I think I already said that though. One example is this. Let's say base C is on one side, base T is on another. Base C would have a G paired up with it, so it would be able to replicate itself that way, and the T would have an A paired up with it. They would be on the same strand half of DNA, but not in the same group. And this is called base pairing. And then the helix, the double helix, is just the blue and red lines you see on this model. Thank you. Goodbye.